Bismillah ar-Rahman ar-Rahim. This is the second lecture in general bacteriology, and we will discuss the bacterial growth. Bacterial growth means increase in the sum of all components of the organism and then multiplication and division. When the bacteria undergo growth, the cell begins to prepare all the enzymes, metabolites, and requirements required for the division and the multiplication, and hence the cell increase in the size. But this increase in the size is not considered growth. But when the bacterial cell is divided into two daughter cells, this is considered bacterial growth. By another word, growth is increase in the number of bacterial cells, not in the size of the cell. When the bacterial cell undergo growth, the one cell gives millions of cells. And when the organism is streaked over this artificial media, we can detect these millions of cells in the form of colony. Each dot here represents a colony. One colony represents millions of cells after growth and multiplication. Bacterial division, as we discussed in the first lecture, is by simple binary fission. The bacteria elongate and duplicate its DNA material. After that, elongation of the cell wall and cytoplasmic membrane inside, and separation of the cell into two daughter cells by simple binary fission. Bacterial growth is described as exponential growth. What is the meaning of exponential growth? When one bacterial cell is divided into two cells, the two cells give 4 cells. The 4 cells not give 6 cells, but give 8 cells, and so on, 16, 32. Constant growth rate by time. This is the exponential growth. Or by another word, steady state increase in the number of cells by time. So bacterial growth described as exponential growth due to its constant growth rate or steady state increase in the number of cells by time, or logarithmic growth. This is the meaning of logarithmic growth or exponential growth. That means steady state increase in the number of cells by time. Another important expression concerning bacterial growth is the doubling time or generation time. Generation time is the time required for one cell to double. The generation time or doubling time is the time required for certain population or certain bacterial cells to double. And it is different from one bacteria to another, but the average in most of the bacteria is about from 1 to 3 hours. E. coli is an example of rapid growing bacteria where the doubling time is nearly 20 minutes. Each 20 minutes, the population is doubled. Each 20 minutes, the E. coli population present is doubled. And so, in short time after streaking of the E. coli on artificial media, the second day we found heavy growth of the organism, and we can detect these colonies because it have short duration time. In another organism as a Mycobacterium tuberculosis, it is described as a slow grower. The generation time in the Mycobacterium tuberculosis, or the organism responsible for the TB, is about 24 hours. That means when one cell to become double, it takes about 24 hours. And so when we streak the organism on artificial media, we can detect colonies or millions of these cells or the colonies of TB after 2 to 4 weeks. So it described as a slow grower. Of course, the generation time will affect the growth rate. Now we know that the bacterial growth means bacterial division and increase the number of cells. Okay, how I can measure the bacterial growth? In order to follow the progress of bacterial growth, it is necessary to have a method to count the bacterial cells. We have different methods of counting. We can take it shortly. Total count and viable count. Total count, that's mean counting of all the population present in our culture, dead cells and alive cells. While viable count means the counting of only alive cells in our culture. When we have sterile culture media, when we transfer certain number of bacterial cells to this sterile culture media and incubate, by time it is expected that these organisms will multiply and undergo growth cycle and give turbidity. This turbidity represents the growth of the microorganism. We measure the total bacteria present here, either living or dead cells, by spectrophotometric measurement. Spectrophotometric measurement estimates the turbidity of the bacterial suspension. All the bacteria present here will scatter the light, either dead cells or alive cells. While in case of viable count, we only count the viable or alive cells. Different methods are available for a viable count. The most popular one is a pure plate method. Pure plate method. In this method, you don't need to know all the steps of the procedure, but just the principle of the procedure. The viable count here by pure plate method, we want to count the bacterial population here. We want to count the number of bacterial cells in our culture. How to count the number of alive bacteria here? We make serial dilution of our culture. Serial dilution, that means different concentrations of our culture. After that, take 1 milli from each dilution in sterile agar and pour melted agar on it. By incubation, we will find the growth of our bacteria. Of course, this growth represents the viable cells, which is undergo growth. Okay. The undiluted culture will show heavy growth that is very crowded to be counted. Okay, the undiluted form and the first and second dilutions. But after that, we can detect the small number of colonies. We can count this number of colonies, and by certain equation, we can reach to the viable count or the total count of viable count. That means alive bacteria present per unit volume of our culture. This is a pure plate method, and so we know that we can count the number of living cells in a certain bacterial culture. I can count the number of the living bacteria present in certain bacterial culture per unit volume. This is the methods of measurement of bacterial growth: total count and viable count. Okay, now we want to construct the bacterial growth curve. When certain known number of bacterial cells is transferred to clear culture media, we can say, for example, 10 bacterial cells per ml. Okay, when it is transferred to clear culture media and incubate, it is expected to undergo growth. And how can we detect this growth cycle? By time, we can make a viable count. So we make a viable count by time. That means each 4 hours, 6 hours, 10 hours, and so on. And we construct the curve by plotting the number of the living cells against the time. We will have this curve, which is called the bacterial growth curve. The curve consists of 4 phases. The first phase is the lag phase. The first phase is the lag phase. LAG, lag phase. Okay, as it's clear, there is no increase here in the number of living cells. This is the number of living cells. no increase in the number of living cells in this phase, which is lag phase. After that, logarithmic phase. Logarithmic phase where rapid increase in the number of bacterial cells occur. Rapid increase here in the number of bacterial cells occur. After that, there is stationary phase. Stationary phase. Here also no change in the number of viable or living cells. Stationary phase. After that, death or decline phase, where the number of living cells is fall rapidly. And there is always persistent cells that is alive and the curve not reach to zero, never reach to the zero. There is always resistant or viable cell by time. Okay, this is a standard bacterial growth curve, consists of 4 phases, and we will discuss these 4 phases in detail. The first phase, lag phase, when the bacteria is transferred to a new media, it takes short time to adapt this new media. It studies this new media. What is the ingredients present? What is the nutritive material it can take? And it began to prepare all the metabolites and the enzymes required for multiplication in this phase. We can detect in this phase increase in the size of the bacteria, but there is no multiplication and no increase in the number of bacterial cells. Okay, this is the first phase, which is the lag phase. This lag phase is different, and there are different factors controlling the lag phase from one organism to another. The most important is the generation time of each organism. After that, the bacteria, after preparing all requirements required for the growth, it undergoes rapid exponential growth. As we described it before, the growth of the bacteria is exponential or logarithmic, and this phase is called the log phase or logarithmic phase. Rapid division and multiplication, and so increase in the viable count exponentially. That's mean by time. At the end of this phase, the bacteria undergo multiplication several times. It began to exhaust all the nutritive materials present in the media. Also, byproduct and the waste material began to accumulate in the media. So, it began to undergo or to reach to the stationary phase, where the exhaustion of nutritive materials and accumulation of toxic product make the growth rate nearly equal to the death rate. Here in the stationary phase, there is multiplication occur. Increase in the number of cells occur. But in contrast, there is death occur due to the accumulation of toxic product. So the final or the net result is no increase in the viable count. Stationary phase here, no increase in the viable count. Although there is a multiplication occur, but in the same time there is a death occur, and so the net result is no increase in the viable count. But in lag phase, as we said before, there is no increase in the viable count, but in lag phase there is no multiplication. In stationary phase, no increase in the viable count, although there is still growth is present or multiplication present, but in contrary, the death is occur also. This is the difference between the stationary phase and lag phase. After that, no nutritive material is present. The pH of the media is changed due to accumulation of the byproduct, toxic product, and so the bacteria undergo rapid decline or death, where the viable count reduce rapidly. As we said, there are a small number of survivals may present for months or even years, and so the curve never reach to the zero. There's also a persistent cells here, so the viable count never reach to zero. Okay, this is the standard bacterial growth curve. The same cycle is occur when the organism enter to the human body. It is the same condition. Microorganism enter to the new environment, which is the human body. The lag phase where no growth, no multiplications occur here, it is the incubation period in the human. When the human when the microorganism or bacterial cell enter to the human body, it takes short time for incubation period where no clinical signs or symptoms appear. But in this phase, as we said before, the organism begin to accommodate the new media or the human body and begin to prepare the requirements for their growth and multiplication. After that, it undergoes rapid growth and multiplication where severe signs and symptoms, fever, and all the symptoms is occur in this dividing phase or exponential growth. After that, the signs and symptoms become slighter or fall in the fever, and other symptoms occur in the stationary phase. And after recovery, this is the decline phase where the organism or the death rate increases, and the convalescence or recovery stage is the decline stage. Now we are going to talk about the requirements for bacterial growth. Bacteria, like any other living cells, require nutritive material for maintenance of their metabolism and for cell division. They are widely different in their nutritional requirements, and they are divided into two main groups: autotrophs and heterotrophs. Autotrophs and heterotrophs according to the nutritional requirements. Autotrophs and heterotrophs. What about autotrophs? Autotrophs are the bacteria live in the environment. They are saprophytes. Saprophytes, that means live in the environment, in the soil, in the water, in the air. They don't need to parasitize in the host. They don't need to live inside human or animal body. They are able to synthesize their own nutritive material. Okay? Autotrophs are able to live in the environment freely without any host because they are able to synthesize all the requirement and nutritional requirements required for their growth and multiplication. And so these free living organisms are of no medical importance. They are called autotrophs. They are bacteria that live in the environment, in the soil, in the plant, in the water, and so on. They can utilize the simple inorganic constituent present. They take the carbon from the carbon dioxide in the atmosphere. They take the nitrogen from the ammonium salt present in the environment and can synthesize their own requirements for their growth. And they are free living and they are of no medical importance. But the heterotrophs, they are the pathogenic bacteria that live inside the human body. And so they are of medical importance because they cause diseases in human body. These heterotrophs, they are unable to synthesize their own nutritional requirements by their own. They require to parasitize in a human or animal cell to take the complex preformed organic substance as sugars, proteins, and other complex organic substances and spread them to take the requirements for their nutrition from this. So the most medical important bacteria that we will study are heterotrophs. They can be cultivated on the culture media by providing these required or complex organic substances or all requirements of their growth in enriched media or in artificial media, and so they can grow and multiply. And we can identify them in artificial media. The second requirement for bacterial growth is the gaseous requirements. And according to the gaseous requirements, first, oxygen. According to oxygen requirements, the bacteria can be divided into different groups. Certain groups require oxygen for their growth. Absence of oxygen leads to their death. They are called obligate aerobes. Obligate aerobes. Obligate aerobes. Their growth is here near to the oxygen, near to the surface, because their metabolism is dependent mainly on oxidative phosphorylation. They require oxygen for their growth. Okay? Absence of oxygen leads to their death. Other group is obligate anaerobes. Obligate anaerobes, that means oxygen led to their death. They grow away from oxygen, deeply inside to be away from the oxygen present here in the surface. They are obligate anaerobes. Other organisms are able to grow in the presence or in absence of oxygen. They are called the facultative anaerobes. They can choose between presence of oxygen or absence of oxygen. Other organisms are microaerophilic. Microaerophilic, the word micro, that means very small, and so microaerophilic, that means they are able to grow in the presence of small concentration of oxygen. Small, so they are near to the surface to take small concentration only of the oxygen, which is optimal for their growth. Again, the bacteria can be classified according to oxygen requirements into obligate aerobes. That is, the bacteria grow only in the presence of oxygen. The most important example, Mycobacterium tuberculosis and Pseudomonas aeruginosa. They are obligate aerobes, strict aerobes. They undergo metabolism, and as a byproduct for the metabolism, there's a superoxide ions are formed. This superoxide ion is toxic to the cell. The obligate aerobes or obligate aerobe bacteria have a superoxidase dismutase enzyme that converts this superoxide ion into hydrogen peroxide. This is the enzyme present in the bacteria, in the aerobic bacteria, converting this toxic byproduct or toxic free radicals of oxygen, superoxide ion, into hydrogen peroxide. Hydrogen peroxide also toxic to the cell, and so the bacteria produce catalase enzyme to decompose this hydrogen peroxide to water and oxygen. Okay, this is a process present in the obligate aerobes. In case of obligate anaerobes, presence of oxygen led to their death because they don't have these two enzymes. They don't have the superoxide dismutase or the catalase enzyme. Obligate anaerobes don't have these enzymes, and so they don't able to get rid of these toxic products, and so they don't go oxidative metabolisms. Other group is the facultative anaerobes. They can grow in the presence or in the absence of oxygen. All the family Enterobacteriaceae are facultative anaerobes. And the last group, according to oxygen requirements, is a microaerophilic that their metabolism depends on low oxygen tension, lower than present in the atmosphere, as in case of Campylobacter jejuni. Carbon dioxide requirements. All the pathogenic bacteria require a small amount of carbon dioxide that is present normally in the atmosphere. But with certain exceptions, bacteria called Procella abortus and also Neisseria require higher concentration than is present in the atmosphere, nearly 5 to 20% of carbon dioxide. But generally, most of the pathogenic bacteria require carbon dioxide that is present in minute quantities, as it's normal percent in the atmosphere. This is a gaseous requirements for bacterial growth. Other requirements for bacterial growth is the temperature. Bacteria require appropriate temperature for their growth. Most pathogenic bacteria that parasitize in the human body, the optimal temperature will be 37 degrees, which is a normal human body. They are called mesophiles. Mesophiles, that means their optimal temperature is about from 25 to 40 degree percent nearly, or the optimal is 37, which is the temperature of the human body. The most pathogenic bacteria is the undergroup and mesophiles. Other bacteria present in the nature from the saprophytic bacteria. A bacteria can grow in cold temperature, 0 or even smaller than 0. It's called psychrophiles. Other types of bacteria can grow in high temperature, reach to 60% as in case of thermophiles, and also other obligatory thermophiles more than 60% present in under the seas and in other environments which is very hot. They are mainly saprophytes, either psychrophiles or thermophiles. They are saprophytic bacteria. As we said before, saprophytes that are live in the environment of no medical importance, but most of the pathogenic organisms of medical importance that cause human infection are mesophiles. Another requirement for bacterial growth is the moisture, as any living organism require water for bacteria require water for their growth. Spores resist dryness for a long time, as we discussed in the first lecture. pH are the acidity and alkalinity. The pathogenic bacteria are neutrophiles. That means the optimal pH is the neutral pH from 6.5 to 7.5 nearly. With the exceptions, Vibrio cholera. Vibrio cholera is the organism that causes cholera. Vibrio cholera is alkaliphiles, that means grow in alkaline pH. Helicobacter pylori is acidophile, that withstand pH, acidic pH nearly 3. Another requirement for bacterial growth is the light. But here, light causes injury or killing of bacterial cell. Most pathogenic bacteria killed by the sunlight because it contains the UV rays, which make mutations in the DNA of the bacteria and lead to injury in the bacterial cells. This is the most pathogenic bacteria, but some of the saprophytes that are present in the environment, as we said before, of no medical importance, contain chlorophyll and undergo photosynthesis and called photosynthetic autotrophs. But the most pathogenic bacteria injured by light. And so when we cultivate the bacteria in the lab, streaking of the organism on a culture media containing suitable nutritive material, pH, neutral pH, the temperature is adjusted in the incubator. Also, no light is entered inside the incubator. That means the bacteria grows in dark, while light make injury in the bacterial cell. Ionic strength and salt concentration. Wide range of salt concentration may be tolerated, but in short, most pathogenic bacteria prefer the isotonic solutions or neutral salt concentration, but some bacteria can grow in high salt concentration or even require high salt concentration, and these bacteria are called halophilic bacteria. Halophilic bacteria, this is a bacteria that can grow or require high salt concentration, for example, Staphylococcus aureus or Vibrio parahemolyticus. Now we are going to talk about another topic, which is the host-parasite relationship. The host here is a human, and the parasite here is the bacteria. Bacteria can be classified into saprophytes and saprophytic bacteria and parasitic bacteria. As we said, saprophytes, that means they are free living in the environment. There is no need to parasitize in hosts. So they are of no medical importance to us. And we are going to discuss the parasitic bacteria. These parasitic bacteria that live in or on the living host, human body, inside the human body or over the skin. They are of two types, commensals and pathogenic. Commensals and pathogenic. Commensals that's present inside our body without causing disease. In some cases, they are beneficial. For example, in the gut, you know that there are millions of bacterial cells in our gut. E. coli, for example, present in the intestine, play an important role in the absorption of certain nutritive materials and in the synthesis of certain vitamins, as vitamin K. Also, there's an organism present on our skin. All these organisms don't produce disease. In normal condition, don't produce disease. They are friendly bacteria that is present in our body or on our body and play an important role and beneficial role in some cases. But you must take attention from these organisms. In certain conditions, under immunocompromisation, if the immunity of the body is decreased for any reason, cancer, viral infection, or any other reason, in pregnant women, in any other reason for immunosuppression, it takes the chance to initiate a disease. So they are called opportunistic. That means they may cause a disease under certain conditions if the immunity of the body is decreased for any reason. Also, it may cause disease if there is change in their normal habitat. Normal habitat, that means the normal site. As we said, E. coli, it is normally present in the GIT, normally present in the intestine. This is the normal habitat. But if it is transferred to another area, if it reaches to the blood, if it reaches to the urinary tract, it will go infection. It will make a urinary tract infection. Okay, so these commensals, yes, they are friendly, they are beneficial, but in some extent they are dangerous, as in immunocompromisation they make a serious infection. Also, if they change their habitat inside the body, they will make infection. The other group is the pathogenic. They are always cause disease. Presence of pathogenic bacteria inside the human body, that means serious infection. The steps of infection to occur. First, the organism enter inside the human body. It must have route of entry, either through the GIT from the mouth, either through the respiratory tract from the nose, from the abrasion in the skin, in the mucous membrane, and so on. Enter to the body first through the entrance, port of entrance, then begin to adhere to the cells and begin to multiply, and then the infections, the signs and symptoms occur, and after that it also exits to be spread to another sites. We have two types of infections you must know. The patient, it is very clear, he is a clinically ill patient. All the manifestation and signs of symptoms all appear with marked tissue damage. But carrier, it's much more dangerous than the patient. Carriers are apparently healthy individuals. Their immunity is very strong. They acquire the organism in subclinical dose, or no symptoms appear, silent infection, no tissue damage, and so they communicate freely in the community and disseminate the bacteria. So the carriers are much more dangerous than the patient. We can isolate this patient, take care from him, but carrier move freely in the community and disseminate the organism as there is no signs or symptoms, no infection appear on him. He is apparently healthy. And this is the difference between the carrier and the patient. Now, what are the factors affecting disease production? When the organism enters inside the body, what are the factors that lead to the presence of infection or appearance of the signs and symptoms or no? The first factors is the host factors, the strength of our body, the strength of our immunity. This will control the infection. But there are other factors related to the microbe itself. The factors related to the host will be studied in the part of immunity, but here we give light about the microbial factors. Pathogenicity and virulence. Pathogenicity, that means the ability of the bacteria to cause a disease. The bacteria is pathogenic or not. It has the ability to cause a disease or not. This is the pathogenicity. While virulence refers to the degree of pathogenicity. And the degree of pathogenicity or the virulence here, it depends on the certain weapons the bacteria have, which is called the variance factors. They are the weapons by which the bacteria can cause severe form of disease or severe stage of the disease. There are different variance factors. Each bacteria have 1, 2, 3 or no of these variance factors. Some bacteria have capsules, as we discussed in the first lecture. Others don't have. Some bacteria have pili, flagella. Some bacteria produce exotoxins. Others have endotoxins, and so on. There are different variance factors, as we said, that are the weapons that enhance the pathogenicity and the ability to cause a disease. These variance factors will be discussed. The first is the adherence. When the bacteria enter inside the human body, some have a specialized structure that help in adherence to the host cell. And so it starts the process, the pathogenesis and disease process. While bacteria that don't have this specialized structure will get out of the body without infection or without the initiation of the disease. For example, teichoic acid that's present in the all gram-positive bacteria. It is the cell wall component in the gram-positive bacteria. Teichoic acid are responsible for the adhesion to the host cell. Also, pili or fimbria, which is present in the gram-negative bacteria, responsible also for the adhesion to the host cell. For example, Neisseria species. Neisseria have pili on its surface. These pili responsible for adhesion to the host cell. Okay? But the mutants of Neisseria that lack these pili are avirulent. That means they don't cause disease. While the strains that have these pili will adhere to the epithelium of the host cells and begin to initiate the disease. Also the slime, which is loosely, it is type slime. We discussed it in the first lecture, capsule and slime, and we said that slime is a loose coat around certain type of bacteria. Certain type of bacteria have slime, as or for example, Staphylococcus epidermidis. This organism has slime around the organism, which is responsible for the adherence on the endothelium of the heart valves, and so initiate the endocarditis. and so on. So the bacteria that have specialized structure for the adherence to the host cell are virulent. And adherence or colonization factors are considered from the virulent factors. The second is the biofilm formation. As we said, the bacteria that have slime around it, this slime consists of the polysaccharide that is attached to each other, and when the bacteria enter to the host cell, begin to attach firstly, and growth, multiplication, maturation, and due to the presence of this slime and polysaccharide, it make layers and aggregations that contain large number of this bacteria. This is called biofilm, and always attached to a solid surfaces as a catheter or pathogenic valves and so on. This biofilm is very, very dangerous as it protects the organism from the phagocytosis and immune cells. Also, the bacteria present inside these biofilms, the antibiotic will not reach to them in the proper concentration, and so they resist the antibiotics. And so these infections will be persistent and very difficult to treat. Okay? So the bacteria that have the ability to form biofilm will make a very persistent and difficult to treat infections. Okay? Another variance factors is the invasiveness of the host cells. The ability of the microorganism to invade the tissue and multiply inside it and destroy this tissue and then spread. Okay? This is one of the variance factors. This invasiveness may be directly by the bacteria itself, that's mean. Here the salmonella. Salmonella is a bacteria that make invasion to the intestinal cell. Here's the invasion by the organism itself. It invades the cells and cause destruction in the cells, multiplication and then spread. Okay, but the other tool of invasion is through certain extracellular enzymes. Some bacteria able to produce enzymes. This enzyme will destroy the tissue, and so it can invade this tissue and spread from it. For examples for these bacterial enzymes, hyaluronidase enzyme and collagenase enzymes. Hyaluronic acid and collagens are the cement substance between the connective tissues and cells. The cells of human body and connective tissue have cement substance between them that make them tied to each other is the hyaluronic acid and the collagen. And when the organism produce enzymes that destroy this cement substance, it will result in invasion of this tissue. Okay? As in case of staphylococci and streptococci, they produce hyaluronidase enzyme and collagenase enzymes which degrades the collagen and hyaluronic acid present in the tissue, and so it enhances the bacterial spread through the subcutaneous tissue and through the body. Other important bacterial enzyme is the coagulase enzyme. Coagulase enzyme produced by Staphylococcus aureus. This coagulase enzyme, and from its name, it makes coagulation of the plasma with the deposition of the fibrin around the region, and so make localization of the organism. Localization of the organism in certain area with a deposit of fibrin wall result in the persistence of infection. Also, this surrounding of the organism result in escaping of intracellular killing and phagocytosis. So these enzymes are considered one of the weapons of Staph aureus to make localized and persistent infection, which is a coagulase enzyme. Another enzymes produced by bacteria, for example, streptokinase enzyme, produced by Streptococcus pyogenes. This streptokinase, or by other word fibrinolysin, make lysis of the fibrin clot. Lysis of the fibrin clot. This enzyme produced by the organism to lysis all the clots formed and all the viscous constituent of the pus and the inflammatory exudate, and so facilitate its spread inside the tissue. These enzymes are used in the treatment of thrombosis. They make lysis of the thrombi in a commercially available product. They are synthesized by fermentation process from the Streptococcus pyogen, streptokinase enzyme. Another enzyme considered one of the important variance factors produced by certain bacteria is the IgA protease enzyme. IgA, or immunoglobulin A protease enzyme. This enzyme degrades the immunoglobulin A, which is present in the mucous membrane as a line of defense against infection by a certain organism as Haemophilus, influenza, and Streptococcus pneumoniae. These strains produce the immunoglobulin A proteases, which destroy the antibody immunoglobulin A, and so eliminate the protection of the host by this antibody. All these enzymes are considered variance factors for the organisms. Another variance factors related to the pathogenesis and virulence of the bacteria is the intracellular multiplication. Intracellular multiplication. Normally, the phagocytic cell, in normal case, this is the bacteria, and when the bacteria enter to the body, the phagocytic cells try to engulf this bacteria first by the formation of this vacuole that engulf here the bacteria in this vacuole in what's called phagosome. Phagosome is the vacuole that's containing the bacteria. The phagocytic cell containing another vacuole containing a lysozyme enzyme. This enzyme that is responsible for the killing of this bacteria. After the phagosome formation, the phagosome and the lysosome is fused together to form phagolysosome, where intracellular killing of the bacteria is occur. This is in normal condition. Some bacteria are resistant to this intracellular killing by inhibition of the fusion of phagolysosome together, and other bacteria can escape from the phagolysosome to the cytoplasm. The first case, the bacteria that prevent the fusion between the phagosome and the lysosome, this is in case of Mycobacterium tuberculosis and Legionella. They are able to inhibit the fusion between the phagosome and lysosome, and so they escape the intracellular killing. Other bacteria, as Listeria, have the ability to escape into the cytoplasm before intracellular killing. Other virulence factors produced by the bacteria is the bacterial toxins. Some bacteria have the ability to produce toxins which are responsible for the pathogenesis. The toxins are of two types: exotoxins and endotoxins. Exotoxins, that means the bacteria produce it external to its body. The bacteria produce or release the toxin outside of the bacterial body, and so it's called exotoxins. While endotoxins is the part or component of the bacterial cell wall. As we discussed before, it is a part of the gram-negative bacterial cell wall, which is the lipid A fraction in the lipopolysaccharide of the gram-negative bacterial cell wall. Again, two types of bacterial toxins: exotoxins and endotoxins. Exotoxins, that means the toxin is released out of the cell. Exotoxin is released out of the cells, while endo, that means inside, it is a part of the component of the bacterial cell wall. In case of gram-negative bacteria, the lipopolysaccharide and especially the lipid A part of the lipopolysaccharide is considered endotoxin. It doesn't cause toxicity except after the lysis of this bacteria cell. When the lysis of the bacteria occur inside the human body, after lysis, the lipid A part is released and so it causes toxicity. This is the exotoxin and endotoxin. And what about the comparison between them? Exotoxin can be released out of the cell either by gram-positive bacteria or gram-negative bacteria. There are many examples for gram-positive bacteria that produce exotoxin, also gram-negative bacteria that produce exotoxin, while endotoxin only in the gram-negative bacteria, as we said, because it is a part of the bacterial cell wall component, which is the lipid A part. And from the name lipid, that means it is lipid in nature. While the exotoxin is protein in nature. While endotoxin is lipid in nature. Exotoxin is protein in nature, so when it is released inside the body, it is immunogenic. That means it stimulates the immune system. Any foreign protein inside the body will stimulate the immune system much more than the lipid, and so it is highly or strongly immunogenic. The exotoxin is unstable, destroyed at 60 degrees nearly, while endotoxin is much more heat stable. Exotoxin is produced by certain organisms, and each exotoxin have specific mechanism of action and act on specific receptors inside the body, and so the action here is very specific. Okay, the exotoxin action is very specific. Each organism produces certain toxins that affect certain site or certain system. For example, bacteria called diphtheria is a gram-positive bacteria produce very powerful exotoxin that affect the heart, for example. Okay, other bacteria as Clostridium tetani produce a toxin affect the CNS. So the action of exotoxin is very specific. While in case of endotoxin, all the gram-negative bacteria have the lipid A, which act all non-specifically. Okay, it causes fever, it causes hypertension and shock by non-specific mechanism. After that, conversion to toxoid. What is the meaning of conversion to toxoid? Only exotoxin can be converted to toxoid. Only exotoxin can be converted to toxoid. It is wrong here. Exotoxin can be converted to toxoid, and endotoxin can't be converted to toxoid. Okay, exotoxin can be converted to toxoid, and endotoxin can be converted to toxoid. Okay, here the endotoxin and exotoxin are replaced. Here in the last slide, the endotoxin and exotoxin are replaced. Here the exotoxin and here is the endotoxin. Again, in this slide, the exotoxin and the endotoxin are converted. Here is the exotoxin and here is the endotoxin. Exotoxin can be converted to toxoid. What is the meaning of that? When we add formalin on the exotoxin, as we said, exotoxin is a powerful toxin. Okay, it causes destruction of many systems inside the body. If this exotoxin is treated with formalin by special technique, it will lose its toxicity. it will become untoxic. But it's still protein, and so it still stimulates the immune system, and hence we can use this exotoxin after treatment with formalin in what's called toxoid. It can be used in the vaccination. As we said, diphtheria produce a powerful exotoxin. We said tetanus organisms produce a powerful exotoxin. Treatment of these toxins by formalin will result in toxoid. This toxoid can be used as a vaccine because it is non-toxic now, but it's still immunogenic and stimulates the immune system. This is the meaning of toxoid. The exotoxin is convertible to toxoid, while endotoxin is not. Finally, the control of exotoxin is, or the genes of exotoxin is present extrachromosomally. The genes of the exotoxin is present extrachromosomally on the plasmid or on the prophage or other, while endotoxin is on the chromosome. We finished the lecture, and we wish you good luck. Thank you.